All right, this video is going to cover the steps in the Krebs cycle. Now, the Krebs cycle is the primary oxidative phosphorylation of all three macronutrients coming into the matrix of the mitochondria using acetyl-CoA and going through this cycle. So again, remember that when we're looking at glycolysis for this unit, the two pyruvates get converted to two acetyl-CoAs, so everything that we go through here, there's going to be two of them. So this first step here, we're taking acetyl-CoA and it's combining with oxaloacetate or OAA. That's combining together to produce citrate. Now, citrate is going to then have an isomer to isocitrate. And one of the things that you can see here that is just a little bit different with the two different molecules is that we now have the OH group on the last carbon. So it was on the second carbon previously. So this is just an isomer. Isocitrate then goes to alpha ketoglutarate, and alpha ketoglutarate is going to gain a double bond. Now, when we gain this double bond, we have to use a dehydrogenase, so we're removing a hydrogen. Now, what that means is we have to have an energy acceptor, so NAD becomes NADH. So this is the first step when we are producing an energy carrier in Krebs cycle. Alpha ketoglutarate is then going to be converted to succinyl-CoA. And you can see here, we're just adding this succinyl-CoA group to the molecule. We have to, again, dehydrate here. So we're using the enzyme alpha ketoglutarate dehydrogenase. That key word in the enzyme dehydrogenase means that we're going to be producing a second energy carrier, or NADH. Succinyl-CoA is the conversion to succinate is going to release GTP, which ultimately converts to ATP. As you can see, we remove the CoA from succinyl-CoA to become succinate. And we use this enzyme succinyl-CoA synthase because we're making succinate. From there, we're going to make fumarate. And as you can see, fumarate here is also going to gain another double bond. And that double bond is going to require us to dehydrate. So succinate dehydrogenase is our enzyme that's responsible for now accepting that hydrogen and producing our third energy carrier. Now this time it's FADH2 that we produce. Fumarate gets transformed to malate. And you can see here the difference with malate is just by putting the oxygen group here. So with malate, we use fumarase to then become oxaloacetate or OAA. And that is going to then get a double bond here, which means that we are dehydrogenating. We're taking away the hydrogen, and in this final step, we're going to produce our last energy carrier, NADH. And that completes the Krebs cycle. Again, that is going to be one molecule of acetyl-CoA going through the entire Krebs cycle, or TCA. Now after this, we're going to go to the inner membrane of the mitochondrial. So if you recall, again, the Krebs cycle is located here in the mitochondria matrix. And then we have these protons, protein complexes 1, 2, 3, and 4 embedded along that inner mitochondrial membrane. And what happens is we have another layer up here as well, which is the outer membrane. So inside of this here, we're going to pump all of these hydrogens from those energy carriers. And ultimately what happens is a gradient of high to low comes through ATP synthase, which is this little blue blob right here. And at the end here, it's going to basically conform ADH to ATP, which is how we produce so many ATPs. So this is just another example, a different picture. Here's your outer membrane. We're in the intermembrane space of the mitochondria. Down below is the Krebs cycle, where we just finished producing those energy carriers. At complex 1, we have NADH come in, and it's going to pump a hydrogen into the intermembrane space. FADH comes into complex 2, which is why FADH produces fewer ATP molecules for each FADH2. This is also going to pump hydrogens, and as these energy carriers continue to go through the complexes, they're going to continue that electron transport and produce all of those hydrogens in the intermembrane space. Now, with all of the protons up in this intermembrane space, we then have a high concentration, and we're going to filter it through this ATP synthase to a low concentration. So these protons get pumped through ATP synthase, where at the end we have ADP and a phosphate group ready to bond to make ATP. So that's exactly what happens when the hydrogens get pumped through, which is why we get so many ATPs produced from the NADH and FADH2. Again, you can test your knowledge to see how many total ATPs are produced from the Krebs cycle and oxidative phosphorylation.